Hello everyone. Today I'm going to be telling you about a project I've been working on over the last year or so on reproductive accessory glands. So when we think about animal reproduction, we tend to focus on two things, the sperm and the egg. And when we consider these two gametes, the most obvious questions that come to mind are about where they are made, how they are made, and how they come to meet. And historically, that's what a lot of the research on reproductive biology has been focused on. But what we've learned is that by focusing in on the sperm and egg, we might miss out on some of the bigger picture and some very important aspects of reproduction. We now know that reproduction is immensely complicated with a lot of moving parts, and there are aspects that are crucial to reproductive success even though they are not directly related to the gametes. One such aspect is the reproductive accessory gland. In general, reproductive accessory glands are defined as an organ involved in the reproductive system that does not directly produce or release gametes. Today, I'm only going to be talking about male accessory glands, and these types of glands are possessed by some mammals, insects, and fish. In some species like humans or Drosophila, we've studied accessory glands pretty well. In human males, the prostate, the seminal vesicles, and the bulbourethral gland could all be considered accessory glands, and we have a pretty good idea about their form and function. Generally speaking, accessory glands usually produce seminal fluid and other substances involved in sperm transport, storage, and performance. And these products can have some striking effects. For example, in Drosophila, some of the proteins produced by the accessory gland are transferred to the female during mating and can influence the female's behavior and even their life expectancy. But in fishes, we know a lot less about these glands, which is in part due to the fact that they are relatively uncommon. However, we do know of a few groups that have them, such as the blennies, gobies, catfish, and toadfish. An example of an accessory gland is shown on the right here. This one is from a species of toadfish, and the accessory glands are shown in orange, just below the testes, which are the large pink structures. But these glands can vary significantly in both form and function. And while some specific functions of these glands have been described in some species of fish, for the vast majority, the function still remains unknown. And fishes represent a really exciting system to study these glands. Fish are the most speciose group of vertebrates, and they display incredible diversity in all aspects of reproduction, making them an ideal system to study the evolution of reproductive traits like these accessory glands. So the goal of my project was to comparatively investigate the evolution of these glands across fishes in order to answer a couple of questions. My first question is, how did these glands evolve? And to answer that, we can look at the evolutionary history. For example, how many times has it evolved independently and in which groups of taxa? My second question is, why did these glands evolve? And to answer this, we can look at whether the evolution of these glands is correlated with other life history traits. And there are a couple hypotheses about what these other traits may be. The first hypothesis proposes that these glands arose as a means to enhance sperm competition, which is the phenomenon of sperm from multiple males competing for fertilization. There is some evidence for this hypothesis, as these glands aid in sperm storage and the production of seminal fluid, both of which can influence sperm performance and competition. But it's a bit tricky to test this hypothesis, considering sperm competition is not a concrete, singular trait. While theory on sperm competition states that males will benefit from increased investment in sperm if there is risk of competing with another ejaculate for fertilization, which is what's shown on this graph here. In fish, the risk of competition is influenced by factors like mating system and spawning behavior, so we can use those traits to gauge sperm competition risk. If this hypothesis were correct, we'd expect to find accessory glands in species where multiple males compete for the fertilization of a single female. The second hypothesis proposes that these glands arose as a means to enhance parental care, and there is some evidence for this as well. In some species, these glands help produce sperm trails, which is this viscous mixture of sperm and other substances which increases the efficiency of fertilization in a nesting scenario. As well, the products of these glands have even been shown to have antimicrobial effects, which enhance egg survival. If this hypothesis were correct, we'd expect accessory glands to be more likely in species that exhibited parental care, specifically ones where the care was provided by the male. It's important to note that these hypotheses are not mutually exclusive, so both or even neither could be true. To actually test these hypotheses, we compiled a large data set on accessory glands and other reproductive traits for 607 species representing 246 families. And we tried to sample all families in the phylogeny that we were using to ensure a good taxonomic distribution. And then we conducted a couple of different analyses to answer the questions we were interested in. To look at how accessory glands evolved, we used ancestral reconstruction through a method called stochastic character mapping. In this method, you can simulate the evolution of a trait to determine ancestral states and how many times a trait was gained or lost. To answer why these glands evolved, we used analyses of correlated evolution. And we actually used two slightly different methods, but the one I will be discussing today is called Pagel's test of correlated evolution of discrete binary traits. And this test tells us quite simply if the evolution of two traits are dependent upon one another. Through these analyses, we can assess the level of evidence for both hypotheses as to why these glands evolved. And now I'm going to walk you through some of our results. So what you're looking at here is a phylogeny showing the simulated evolution of accessory glands across our 607 species. The lineages that are red have accessory glands, whereas the lineages that are blue do not. And we found 116 species with accessory glands, representing 26 different families. And you can see on the phylogeny that most of these species are found in three clades, these large clumps of red. These clades are the catfish up top, the gobies to the left, and the blennies to the right. And you can see a few small groups as well, and a few individual species that have accessory glands. And through this simulation, we were also able to estimate that accessory glands evolved likely between 14 to 18 times, and they were lost between 7 and 11 times. The next thing we looked at is the correlations with other reproductive traits, starting with parental care. And the first thing we found is that accessory glands are more common in species with male care compared to species without male care. As you can see here, where I plotted the proportion of species with accessory glands for both groups. And this suggests that there could be a correlation between these traits, but of course we need to control for phylogeny. So this is probably a more accurate representation of the correlation. Here I plotted two trees next to each other. The one on the left is the same accessory gland tree I just showed you, just in a different orientation. And then the one on the right shows you the simulated evolution of male care, which is shown in red. And you can see that the red areas line up pretty well between the two trees, indicating that species with accessory glands are pretty likely to also have male care, and vice versa. And this is a good indication that there is a correlation between the traits, and that was confirmed by Pagel's test. One way to get more detail about the nature of the correlation between these two traits is to look at the transition rate estimates from our analysis. And looking at transition rates is more informative than just looking at the proportions I showed earlier, since the transition rates can actually tell you how fast or how likely accessory glands evolve in different lineages. Here I plotted the transition rates towards the evolution of accessory glands, and you can see that lineages with male care have a much greater transition rate, indicating that they are more likely to evolve accessory glands when compared to lineages without male care. And now we're going to switch gears to our other hypothesis regarding sperm competition. And we investigated sperm competition using a couple of different traits, but the one I'm going to talk about today is mating system. And there are lots of different types of mating systems, so we chose to simplify things and categorize them based on the relative risk of sperm competition. In low-risk systems, females only mate with a single male, and this includes monogamy and polygyny, whereas in high-risk systems, females mate with multiple males, hence the greater risk of sperm competition, and this includes polyandry, polygyandry, and promiscuity. And what we found is that accessory glands are actually more common in species with low-risk mating systems, which have a much greater proportion. And our analysis confirmed a significant correlation between accessory glands and mating systems. And if we look at the transition rates, you'll see a much greater rate from low-risk lineages, which directly contradicts the predictions of our sperm competition hypothesis. Okay, so now let's take a step back and think about our overall questions, starting with how did accessory glands evolve? And we now know that they evolved several times independently and are present in over 100 species. Also, the species that possess these glands are pretty diverse. They are not restricted to any geographical region, or to saltwater or freshwater, or to any particular niche. And even though we only know details for a handful of species, the form and function of the glands in these species seems to be diverse as well. Which brings us to our second question, why did they evolve? And the answer seems to be linked to parental care, since there was a very strong correlation between those two traits. In terms of sperm competition, accessory glands were associated with low-risk mating systems, which goes against our sperm competition hypothesis. And while this doesn't completely rule out the possibility of sperm competition influencing the evolution of accessory glands, this is a pretty interesting finding, since in other taxa like insects and mammals, sperm competition seems to be the driving force behind accessory gland function, but in fish it seems like that might not be the case. However, I don't think we have quite enough evidence to say that parental care specifically has driven the evolution of these glands. While there is a very strong correlation there, accessory gland evolution may be due to a different driving force that is also related to parental care. Other factors could have initially selected for accessory glands, and then parental care specific functions may have evolved from there. The fact is that we're not going to know specifically what drove accessory gland evolution until we describe the functions of these glands in many more species. And conveniently, we now have a list of 116 species that people might want to look at. So these results are pretty cool, but you're probably wondering, why does this all matter? Well, first of all, it fills a clear gap in our knowledge about accessory glands, since this is the first large-scale comparative analysis across fish, and it answers some pretty basic evolutionary questions about how and why these glands evolved. Secondly, it adds to our understanding of reproductive traits in fishes and the kinds of selective forces involved in the evolution of reproductive traits in general. And finally, there could be actual applications of this type of research down the road. If we think about species that possess accessory glands, there are a lot of species that are of scientific, ecological, and economical importance, as well as invasive species and endangered species. Understanding the reproduction of these species could prove useful in many different fields. So thank you all for listening to my presentation, and a big thank you to all these people for making this project possible. I'm happy to answer any questions you may have.